Alright, so in this video, I'm going to discuss hydrogen bonds and how they contribute to protein and DNA structure, and how sometimes they don't contribute as much as we may think. So here's a general hydrogen bond. You've got a donor atom, which is not the hydrogen atom in specific, but rather the electronegative heteroatom that is attached to. This electronegativity, or differential electronegativity, produces a dipole, indicated by this arrow here, where the head of the arrow is the positive end. And that donor hydrogen complex associates with an acceptor atom, which has a partial negative charge, which is usually due to a localization of electrons in one location. So this is a typical dipole-dipole interaction, which you're very familiar with. You're probably very familiar with the energy diagram too. Modeled as such, this is energy on the y-axis, distance r on the x. I'll use the specific case of a nitrogen associated hydrogen and an oxygen for this, and this r is measured between the hydrogen and the oxygen in this case. So if we do that, the location of most stabilization is about 2 angstroms, and the amount of stabilization is about negative 10 kilojoules per mole. So this number, negative 10 kilojoules per mole, importantly, is derived from general gaseous studies, which are usually conducted in a vacuum. So, as we know, biological systems are not conducted in a vacuum. So if we want to look at how hydrogen bonds are going to actually stabilize and interact in a biological system, we have to take into account the effects of the major solvent of biological systems, which is water. So right here we have a little example of 2 acrylamide molecules associating with each other via hydrogen bonding. And we're going to do a little mini thermodynamic analysis to see what the stabilizing effect of the hydrogen bonds, the hydrogen bonds between the acrylamide molecules are. So let's specifically take a look at this hydrogen bond, which is analogous to this hydrogen bond over here. So let's say that this hydrogen bond on the left side has a stabilization of, as we discussed before, about negative 10 kilojoules per mole. And then let's say that between the acrylamide molecules, it's about the same. So if we want to actually see the stabilization effects of this hydrogen bond, we're going to have to look at the delta H, which, since they're both about negative 10 kilojoules per mole, is about 0. So the idea is that when in water, hydrogen bonds actually don't stabilize structures much. They're useful for a lot of things, but they won't be the primary factor in protein structures, or for that matter, the formation of DNA structure, double-stranded DNA. So one more aspect about hydrogen bonds in general that is often overlooked is the fact that they have these very specific geometries. So in this figure here, we have brought together a bunch of crystal structure data from the protein data bank, and what they've done is they've taken a look at individual side chains, or rather functional groups of side chains, and examined where the most common location of hydrogen bond acceptors and hydrogen bond donors with respect to them are. So in the specific case of the carboxylic acid functional group, you've got the acceptor right here, and the most common location of donors when looking across all the crystal structures there are these indicated by these mesh sites. So if we take a look at the arginine over here, you can see the most common location of hydrogen bond acceptors are specifically here. So if you take a look at the arginine, it's what you would expect. It's this sort of collinear location for the acceptor. But if you take a look at the aspartate or carboxylic acid functional group, you don't have this collinear location. Instead, you've got them sort of splayed out here. And if you remember from general chemistry, you know that is because this is an sp2 hybridized oxygen. And as such, the concentrations of electrons are in these orbitals out here in a trigonal planar orientation. So let's take a look at another one. Here we've got serine here. Serine here is sp3 hybridized, and as such it has this tetrahedral geometry. And because of that, the locations of electrons are roughly here and here. And so, since the electrons are what are actually acting as acceptors, that is the location of the donors. So the idea I want to get across is that hydrogen bonds have very specific orientations. And they are not readily interchangeable between each other. So if you were to take a hydrogen bonding group here that had this carboxylic acid formation, and you were to interchange it with even another hydrogen bond accepting functional group like the one in serine, they would not likely actually have the same orientations. So they're not interchangeable. And that differs from hydrophobic groups, because you can just interchange any alkyl chains and they don't have the van der Waals forces don't have specific orientations required. So if you expand upon the fact that they're not interchangeable and think evolutionarily, you can say that they are not robust to mutations because of this. That is, if you have a hydrogen bonding group that has a specific function and you change it with another hydrogen bonding group, it is not likely that it will have the same orientation, and thus it is not likely that it will lend the same structure to the protein or lend the same catalytic activity. So it is not robust to mutations because of this, the geometric constraints. All right, now let's look at the contribution and necessity of hydrogen bonding when creating protein structures. Specifically, we'll look at the hydrogen bonding that is required by the protein backbone. So the backbone is made up of all these amide groups and carboxyl groups that act as donors and acceptors. The amides are good donors, and carboxyl groups are good acceptors. So if you want to create a regular structure or remove the protein from an aqueous solution, you must satisfy these hydrogen bonds, or else there is a large energetic penalty, and it would not likely to be stable. So there are several ways of satisfying these hydrogen bonds. One of them here on the left is the formation of a beta sheet. So what we have is on the left is a random coil, and here is a beta sheet formation. So in the random coil formation, you have the hydrogen bonding requirements of the backbone satisfied by solvation by water. But what you can do if you want to create a regular structure is make it so that there are hydrogen bonding between two separate backbones. And that's an important aspect of beta sheets, is that they require at least two separate strands. Another way you can satisfy hydrogen bonding requirements for the backbone is to form an alpha helix. So once again, we have the random coil here on the left, and then on the right we have the alpha helix, which forms internal hydrogen bonding. That differs from the beta sheet because it does not require more than one peptide strand. So you can form a stable structure, secondary structure, with only one peptide strand. When these are in an aqueous solution, there is not really an energetic benefit to forming these secondary structures. So secondary structures are actually unstable in water. But let's take a look at what would happen if we were to try to remove them from water and put them into a plasma membrane. So here we have a random coil. Let's take a look at one hydrogen bond to the backbone, and let's insert it into a plasma membrane and take a look at the thermodynamics of it. Same random coil here, insert in a plasma membrane. There's no longer this hydrogen bonding occurring, and so if this has a stabilization energy of the negative 10 kilojoules that we discussed before per mole, this would now have, we could say it's a 0. So there is this energetic penalty to the insertion of a random coil with unsatisfied backbone hydrogen bonding of about, you could say, 10 kilojoules per mole. This varies a lot, so I would not take these numbers as fact. But the idea is that there's an energetic penalty to removing a random coil protein from an aqueous solution, and that energetic penalty is termed the desolvation energy. And that will preclude the insertion of proteins into plasma membranes. But we know that there are, in fact, proteins in plasma membranes. So what actually occurs is you can satisfy those hydrogen bonding requirements by the formation of these regular structures. So an example here is the formation of an alpha helix. And this is now, once again, negative 10 kilojoules per mole. And so the overall reaction, the change in energy, does not preclude, sorry, that is a 0, does not preclude the insertion of a protein into the plasma membrane. So once you get this alpha helix formed within the plasma membrane, you can imagine that the reverse reaction to go back to a random coil formation and remove the hydrogen bonds from the backbone would be unfavorable. And because of that, these secondary structures are stable, very stable rather, in the plasma membrane, whereas they are unstable in water. So now let's take a little bit of a more in-depth look at the hydrogen bonding within these two secondary structures. Here we have an alpha helix on the left. We'll look at that first. There is a definite pattern to hydrogen bonding in the alpha helix. So here's the C terminus. Here is the, this is the N terminus. And this is the C terminus. You can tell by this arrow running through the middle. Always go N to C. And we could say that this is the ith residue, or this is the ith alpha carbon. And this right here is the carboxyl to the ith alpha carbon. You can step up the alpha helix, I plus 1, I plus 2, I plus 3, and then we get to the I plus 4th residue, and the amide on that is what is hydrogen bonding with the carboxyl of the ith residue. So that is important to know, important to remember. Now let's take a look at the hydrogen bonding in a beta sheet. Right here we have an anti-parallel beta sheet. You can tell here's an N, and then it goes N to C here, and then N to C here. And these are associated with each other, fully satisfying all the hydrogen bonding requirements internally within the beta sheet. Internal satisfied, but if we take a look at the edges, such as here and here, we can see that the outer hydrogen bonds are unsatisfied. So this would not be able to be inserted into a plasma membrane unless you manage to bring this outer edge over to this edge here, and that would be the formation of a beta barrel. That is why you're more often to see alpha helices spanning the plasma membrane than beta sheets, because you have to form these large beta barrel structures in order to satisfy the outer hydrogen bonds. The final thing I want to get across about hydrogen bonding with regards to biopolymers is that hydrogen bonding is actually not the primary stabilizing force for the formation of double-stranded DNA in an aqueous solution. And let's go into depth a little bit for that. So we know from many studies that double-stranded DNA is very stable in water. Okay, so there has to be some sort of intermolecular force that causes this to occur. So we can hypothesize that these hydrogen bonding between the complementary strands is the cause of that. But if we think back to the formation or the lack of stability of secondary structures, and if you think way back to when we were talking about acrylamide, you'd know that even if you unzipped the double-stranded DNA, you could satisfy these hydrogen bonds by just solvating them with water. So because of that reason, we know that the delta-delta H, due to hydrogen bonding, is actually very small for the formation of double-stranded DNA, and is not the primary factor. So what the primary factor is actually something called base stacking, where you get this complementary orientation of partial positive and partial negative charges on the bases themselves. So yeah, that is a general overview of how hydrogen bonding contributes to structure in DNA and mostly protein. And hope it was useful for you.